Chapter 19, The Evolution of Populations. So in this chapter, we'll take a look at the processes which allow populations or groups of organisms to adapt in order to fit to their environment. There are so many forms of living things, and if we think about it, they all evolved at one point from a single common ancestor into the great diversity of life that we know today. So let's start talking about population genetics. We know that individuals do not evolve. Individuals themselves do not evolve. But we know that populations can evolve. Now, when you hear the term population genetics, you probably think back to our last chapter when we talked about Darwin. But the important thing to know here is that Darwin did not know anything about Mendel's work on crossing and traits, or didn't know the basis of genetics, which is the science of DNA. So population genetics really became incorporated into the theory of evolution, which we do know Darwin for, not until the 1940s. And this was after the beginning of something that came about, which is called modern synthesis. So modern synthesis is the understanding that there's a joint role of natural selection and genetics in evolution. Now here are two terms. We have microevolution and macroevolution. So we know evolution is change. Microevolution says that a population can change over time. And we see that routinely, populations shifting. Macroevolution says that occasionally there is significant enough change that a whole new species arises. So population genetics is the study of what changes allele frequencies in populations throughout time. Now, an allele, remember, is a version of a gene. So if you have a gene, say for hair color, you might have several different alleles for that. You might have blonde, brown, black. So those are different versions for that particular gene. And the number of those copies or the representation of those alleles in the population can fluctuate, can change. So we have alleles, remember we said those are the versions of a trait, and we have allele frequency, which is the number of copies of those different versions. We also have to consider the gene pool. So the gene pool, if you remember back from last semester, is all the different possible alleles for that trait. And then there's something that's called the founder effect, and we're going to talk about that more in a few slides. That says that there's something that could change the allele frequency in only a portion of a population that gets isolated from the main population. So in the early 1900s, two scientists, Hardy, who was an English mathematician, and Weinberg, who was a German physician, came up with the Hardy-Weinberg principle of equilibrium. And this states that the frequency of alleles is stable from generation to generation if you use the following mathematical equation. Now, that mathematical equation is based on some variables. The first is called P, which is the frequency of a certain allele for a trait. Q, which is the frequency of another allele for that same trait. And that if you look at these two frequencies, we're going to just assume that there's only two versions. If there's more, it gets a little more complicated. Then we can see that P, the frequency of one of the alleles plus Q, the frequency for the other type of allele, always has to add up to 1. So these are percentages, and so when you have percentages, they always have to add up to 1. So let's take a look at our top box. So here we are looking at the allele frequency for yellow colored seeds and green colored seeds. So we have a dominant gene of allele, which is the big Y, and we have a recessive allele, which is the small y, which codes for green. So if we add up the number of copies, so here we have 245 individuals that have this genotype. So obviously they have 2 copies of the big Y. So that would give us 490 big Ys. Plus we have this number of individuals that have 1 copy of the big and 1 copy of the little. And we have 210 of those individuals. So we have 210 big Ys, but we also have 210 little Ys. So we're going to put them in this category. And then lastly, we have the homozygous recessive. So we have 45 of those. If we double that, that means, because we have 2 alleles in each individual, we have 90 alleles for the green seed trait. So we add those all up. 490 plus 210 gives us a total of 700, and 210 plus 90 gives us a total of 300. So we have a total number of alleles of 1,000 in the gene pool. If we want to look at the allelic frequency for the big Y, which is the yellow allele, we would take 700 divided by 1,000, so this is how many big Ys we have, divided by the total number of alleles, and we would come up with 0.7, that's our P. And if we do the same for our little allele, 300 little Ys divided by 1,000, that's 0.3, that's our Q. So now if we add P plus Q, 0.7 plus 0.3, we see that adds up to 1. So we have satisfied and completed the Hardy-Weinberg equilibrium for the allele frequency. Now if we want to look at the frequency of genotypes, so remember genotype is the 2 alleles, we can do a similar kind of thing, and we see that there's a slightly different equation, and that's P squared plus 2PQ plus Q squared has to equal 1. This is for genotypes. This is just the frequency for allele. So now we're looking at frequency of genotypes. So in this particular case, we want to take our genotype frequency, so that's shown up here, our genotype frequency, and we get that by squaring the P, and we get to 0.49. For our genotype frequency for this one, we just multiply P times Q, so 0.7 times 0.3, and so we have 2 of those. And then for our recessive, homozygous recessive phenotype, we will square the Q. So now we see that if we take the 0.49 plus 2 times the PQ plus Q squared, we end up with 0.49 plus 0.42 plus 0.09 equals 1. And if you do that math, that equals. And so this gives us an estimation of what we would expect the alleles to be in this population. But the question here is that populations are always changing. So if this held firm all the time going forward, then we would say that this population is not evolving. It could be this right now, but it might be different in 1,000 years, 1,000,000 years, etc. So if we do see the allelic frequency or the genotype frequency changing, that means that Hardy-Weinberg is not held firm. That tells us that evolution is in fact occurring. So, for evolution to occur, we know the allele frequencies have to change. How does this change happen? Well, there's a number of different mechanisms. One mechanism is called genetic drift. We have two examples. One's called bottlenecking, and the other's the founder effect, which we alluded to before. Gene flow, environmental impacts, mutation, non-random mating, and natural selection. So let's take a look at these different types. So genetic drift says that a particular allele could be eliminated from a population purely by chance. Purely by chance. So here's a figure from our book showing a population of rabbits with different color coats. The B is for brown, and it results in a brown coat. The little b is for white, and it results in a white coat, but only in the recessive condition. So now, if 5 rabbits mate, if only these 5 rabbits happen to mate, we see that there's a reduction in the number of genotypes with the white coat. And if in our next mating that there's only a by chance only mating of these 2 homozygous dominant, then we see that all of a sudden we have completely lost the little b allele from this population. So this is loss of allele due to chance. One example of that is called bottlenecking, and bottlenecking happens when a chance event or catastrophe allows only certain individuals to be left in the population. So that would greatly remove a whole bunch of alleles. Now, what's a chance event? Something like an earthquake or a flood, which will remove a large number of individuals from a breeding population and cause a limitation in the number of alleles that are present. Oops. Another type that I don't have a slide for is that founder effect. So the founder effect is when there is a grouping of individuals within the population that leaves the population and starts a new breeding population. So therefore, usually they leave because they have similar traits, and so they go off and they start a new breeding population, and that usually has a much increased limitation of the allelic frequency. Very, not as many different kinds of alleles. Another way we can change the allelic frequency is called gene flow. So this is when individuals go back and forth between different groupings within a population. So here we have a grouping of brown beetles, and here we have a grouping of green beetles. They're all still in the same species. These are just variations. And maybe one of them comes over here for a season, so we see a change in the allele frequency of this group. And then maybe the next season, some go back, some green ones go over to the brown grouping, and so we see a back and forth flow. So it's not a permanent leaving like we see with the founder effect, but it's a back and forth flow that temporarily changes the allelic frequency. Another thing that can change the allelic frequency is environmental impacts. So certain components of traits might be impacted by conditions in the environment. For example, this alligator, the sex is determined by temperature. So if the eggs are incubated at a lower temperature, you get more females. If the eggs are incubated at a higher temperature, you get more males. That is restricting the alleles in the population, and that has to do with the temperature. So we have something like climate change, which all of a sudden increases the temperature of the environment, then that can have an impact. Climate change can also impact the amount of water in the environment, whether it's dry or really wet. And so this can have an impact on what alleles are going to be present. Another thing that changes how many copies of alleles we have is non-random mating. So this can happen in the wild if individuals selectively choose mates based on some particular phenotype. So if there's a collection of females that happens to like the coloration on a certain group of males, then they're picking based on a particular phenotype, and that's a non-random mating. It's not the case that, any female will mate with any male. Now, this also happens in artificial selection. We know that that is when humans intervene and purposely cause only certain individuals to mate with other individuals. So I have this lovely golden retriever up here, and this golden retriever breed has been impacted by artificial selection. So over years, people have bred golden retrievers for certain traits. And unfortunately, because they have limited the allele frequency by doing that, by artificially selecting, they have also kept in the population a good deal of health problems like hip dysplasia, which causes problems with the hind end of the animals. Now, mutation. Mutation is another way that we can affect the alleles that are present. So mutation, or the changing of an individual's DNA, does occur, but it does occur all the time. It's happening to you and me. But in order for that to impact the species, to cause evolution in the species, that mutation has to happen in the germ cells. Has to happen in the germ cells, in the eggs or the sperm. So now it can happen in a population because good changes may persist. Bad changes or bad mutations may cause individuals to be removed quickly because those individuals don't survive to reproduce. But remember, it's not the individual that's evolving. It would be the entire species, and in order for that to happen, the change has to be in the germ cells itself. So mutations must accumulate within the population and become the norm before we actually see evolution of the species. Now, the last way that we see evolution through allelic frequency changes is natural selection. So natural selection, remember, natural selection is that some traits which appear randomly will allow certain individuals the ability to survive, and because they survive, they can go on to reproduce and pass their genes on to their offspring. So we have three different examples of natural selection. The first is called stabilizing, then there's directional, and then there's diversifying. So these are three different examples. And we're going to look at distribution of phenotypes. That's how a trait appears in a population. So in each of these, you're going to see a blue line and a red line. The blue line is the original population where we see a variety of alleles. The red line is where we're going to limit the number of alleles after natural selection has occurred. So here we're looking at robins, which are a type of bird. They typically lay 4 eggs, and 4 eggs is the population after natural selection. So how did they get to that point? Well, there was a time when you had some robins who laid fewer than 4 and some that laid more than 4. And what happened was those were not beneficial. Fewer than 4, it was likely that maybe mom would step on them and crush them, and you'd have fewer survive. more than 4, then when you have that many chicks hatch, there's less resources for them, less likely for them to survive and not go on to reproduce. So 4 seemed to be the sweet spot, and that's called stabilizing selection, where you see the population shift to a middle of the road phenotype. Directional selection, you see a shift to having one rather than multiple type phenotypes. So in this particular case, we have the original population that had a kind of speckled look to them. Now, just randomly, some individuals would be born with a more darker coloration. And if the environment changes that suits the darker population, like pollution happened in England in the 1800s, then what happened was you saw that these were not as likely to survive. So hence you see a shifting to more of one type of phenotype. Now, diversifying selection means you actually shift from a single population, which would be these white bunnies, to phenotypes which look fairly different, but not like the original. So you get ends of the spectrum. And so what happened here was in these Himalayan rabbits, the rocky environment was not really beneficial to the white phenotype. It was more beneficial to a total dark coat or a mottled coat with some white and some dark. So that's diversifying. So remember that natural selection acts on individuals because some die to affect a change in a population, but individuals do not evolve. Only populations can evolve. Here are some examples of adaptations of natural selection. So these are varieties of natural selection. And this one is called frequency dependent selection. So here we see that the number of individuals with a particular trait will go up or down, and this happens within the population. So this is an example of a type of lizard. This lizard has 3 varieties or phenotypes for the color of the throat. So there's orange-throated, blue-throated, and yellow-throated. And coupled with this coloration is that the orange-throated males are bigger and stronger. The blue-throated males form tight pair bonds with their females. And the yellow-throated males are smaller than either the blue or the orange, but they look more like the females, and so they can sneak in and hang out with the females. So what happens is that if you have an orange-throated population, that's the majority, but a few blue-throated males are around, then they are going to form tight pair bonds with their females and the females won't let the orange-throated males in, even though they're bigger and stronger. So over time, you may see a shift from the orange-throated to the blue-throated males being more represented in the population. Now, if you have a population of blue-throated males, you might have a few yellow-throated males around, and these ones look more like the females, so they sneak in to the females and they can get in there even though the blue-throateds are tight pair bonding because they look like the females, and they kind of sneak in and mate with the females before the blue-throated ones notice. So then over time you'll have a shift to more yellow-throated males. Then with these yellow-throated males, you might see some orange-throated appear in the population, and they're bigger and stronger, and they're going to take these females away from the yellow-throated. So you kind of go through this population. It's kind of like a rock, paper, scissors thing where you have a fluctuation. Sexual selection occurs, and this comes through natural selection, where you end up seeing differences between males and females, and this is because if you have differences that within your males and females, then there might be a higher chance of breeding between particular males or particular females. So in this case, you might have a brightly colored display in your male peacocks that attracts more females. And so over time, you see that more of the males who survive are breeding with females that allows this trait to be carried on. Here we see that the female spiders are bigger than the male spiders. This bigger female means that she can give more resources to her eggs, and therefore we see bigger females over time. And here we see the coloration differences between the male and these female ducks, and that again is attracting female mates. So keep in mind that evolution is more than just natural selection. There are other forces at play that we have mentioned that will change allele frequencies. And lastly, we know that evolution has no long-term purpose or direction. Evolution can go up or down, make changes in good or bad ways. So seriously, there's no long-term purpose or direction. And I really mean it, and you may see this on a quiz or a test. So keep this in mind. Thank you.